Probably the most difficult thing is to be the last speaker in such a long panel. And I believe that in a couple of minutes a new session will start here. I have many things to share. If the chairpersons allow me, let me go through them very fastly, because all of my lecture is actually published in the publication of the meeting. I will go through them, because otherwise I will not have time to go through. We have the intermission. 14:45 there is a new meeting. Congress Director. Let's finish it in 15 minutes. We have 15 minutes. Yeni konuşmacı.
When we look at the issue from the point of pharmacology, if we have selected the right medication and if we give it at the right dose for the correct period, and if the intervals are correct, then we expect some benefit from that drug, even though the individual has not met the medication before, and if he does not respond to the drug, then we can speak about pharmacoresistance. We have to prepare a treatment protocol depending on the characteristics of the individual. Many speakers before me spoke about different factors related to the patient, and such as the age of the patient and other characteristics, and here I will be talking about polymorphisms and genetic differentiation. Patient-related factors and disease-related factors may create differences and may impact the response, and of course there are other concomitant diseases. Medication may be beneficial to patient A, but it will be totally ineffective for patient B, and it will create adverse effects in patient C. Pharmacogenetic profile shows that sometimes we can also have adverse drug effects or undesirable side effects. Pharmacodynamics and pharmacokinetics are two sides of the same subject. We have already heard about the pharmacokinetics, so I will be speaking about pharmacodynamics. Since we are talking about psychiatric medication, we have to speak about monoamines and their transmitters. We have to look at changes at the level of neurotransmitters. We need to look into transduction mechanisms and differences in these mechanisms, blood-brain barrier and proteins in the barrier, plasticity of the nervous system, and of course immune system and cytokines are some of the components. The whole issue becomes highly complex. You know all these pumps, noradrenaline and serotonin pumps. These are two neurotransmitters, and different ions enter the cell, modify the electrical charge. Monoamine concentration is altered, and presynaptic homeostasis is preserved through these pumps. Certain drugs that we give, and sometimes some drugs that are abused, bind to these carriers, and that will alter the functions of the pumps. Here you see some of the carriers, and there are many things that we use in the practice of psychiatry. Carriers may have polymorphism, that's important for us. Noradrenaline transported gene may go through mutation which lead to depression. And polymorphism of serotonin may lead to impulsive behavior, suicidal tendencies, and alcoholism. These carrier genes may be related to the receptors which are found in the disease itself. The whole thing becomes even more complex. Enzymes that we find in the metabolism of monoamines must be taken into consideration. Cytochrome P450, isoenzymes, etc. But we have to look into monoamine metabolism. These enzymes are also important in the whole process. There are many such enzymes. You're familiar with these enzymes? They exhibit genetic polymorphism, and they are important in determining the efficacy of the treatment. And let's first look at TH. That is held responsible for schizophrenia. TPH is related to aggressive behavior and suicidal tendencies. And TPH2 is an indication of major depression. AAD is an important enzyme. There are two different subgroups. One of them is related to bipolar disorder, and the other is related to schizophrenia and ADD. DBH, dopamine beta hydroxylase, is an enzyme that is being studied very carefully. Cocaine-related paranoia, psychotic symptoms of schizophrenia are related to DPH. Monoamine oxidase, there are two subtypes. MAO-A has polymorphism. MAO-A activity increase is related to alcoholism, panic disorders, and major depression. A lower level of the enzyme may be related to schizophrenia and mood disorders. Here you see people who have the same genotype but who have different early experiences and the relationship to their antisocial behavior in adulthood. No matter what the genetic structure is, early traumas, abuse will alter the personality of the individual and will lead the individual to be an antisocial being. So genetic and environmental factors have to be taken into consideration together. KOMT are very important enzymes as well. Schizophrenia, bipolar disorders, unipolar disorders, obsessive compulsive disorders, drug abuse are related to KOMT. Within the same organism, there are endogenous receptors, and there is also intercellular mechanisms. And we give the patient synthetic substances to create different effects. Genetic factors may alter the response that we expect from the medication. One of the most important factors here is intercellular genetic modifications, and what else is happening within the cell. There may be modification at the receptor level. A drug molecule must go and bind to a receptor in order to exhibit some effect. So number of receptors on the cell membrane will increase or decrease the effect of the medication. In chronic drug administration, internalization of receptor molecules will lead to less effective drug activity because the number of receptors are decreased. Receptors are metabotropic or ionotropic. Ionotropic receptors are easy because there is an ion channel and they will modulate the ions and allow a cascade to be triggered. But receptors which have gene protein, the mechanism is even more complex. There is a modulator protein that is bound to the receptor, and the subgroups of this protein are differentiated, and they will modulate the efficacy of the other receptors. Any change in G protein will not change the receptor, but will have an impact on the response we obtain from the medication. So the receptor and the single substance are at the beginning. So starting with G protein, within the cell, the whole process is amplified, and the final response is only possible when the steps are not modified. Gene protein and gene protein related modifications will have an impact on the response that we obtain from the drug. There are many studies that are ongoing. In anxiety disorders, G protein has been carefully studied. In after chronic medical treatment, there could be an effect of GABA-A receptors. Benzodiazepines will not lead to any modification in the G proteins, but buspirone is a partial agonist of 5-HT1A receptors, and when you give this drug in a chronic manner, there will be a decrease in G protein levels, and naturally the treatment is not successful due to the decrease in the level of G proteins. A similar structure can be observed in autism and autistic disorders. Bipolar disorders have been studied for G proteins in the frontal cortex. G protein levels are increased in bipolar patients, and with lithium treatment, the G protein subgroups are also decreased, and all these are important in responding to the medication. G protein is also decreased in depression patients. Tricyclic antidepressants have not led to any change in the level of G protein, but fluoxetine, a ligand of postsynaptic 5-HT1A, will decrease the level of G protein in different parts of the body, and the efficacy of the drug is decreased. Schizophrenia is also studied, and here G protein is studied very carefully. Different levels of G protein have been studied and found to be limited in schizophrenia patients. Post-mortem studies indicate this as well. Dopamine D2 receptor is also parallel to G protein levels. Now we know that receptors and what happens after the receptors has been explained. Let's assume that everything is functioning very normally, so then the drug will have to go through the membrane. The drug molecule will go through the molecules, and naturally it will have to do this through the activity of the transport proteins. There are about 500 to 1200 gene codes, and 15 of them are most important. And maybe the most important for us are protein related to multi-drug resistance protein. There are many subgroups. It's found in the barrier. This protein, there are many varieties of this protein, and it also can be found in peripheral regions. We find them on barrier, on the luminal surface of endothelial cells, and they control the drug interaction. P-glycoprotein has been carefully studied for drug transport. It also has affinity to many chemical materials. It controls many chemical materials, absorption, distribution, of the drugs are regulated by P-glycoprotein and similar glycoproteins. It has an effect on kinetic effect of the medication. It is also an interaction with GP enzymes, so it's very important for us. Today in our treatment principle, especially in refractory patients, we have to take into consideration the PGP inhibition. PGP inhibitors are now being carefully studied. While all this is happening, our brain and our nerve cells are going through a transformation. Until recently, we thought that neurons and the brain were static, but we now know that they are changing. They're going through transformation. The number of synapses increases in time. Sometimes there is a misconnection of synaptic structures which will lead to different pathologies. Neuroplasticity is a result of neuron gene expression, and it controls many changes within the cell. And here we have to add neurotrophic factors. The number of the neurotrophic factors are increasing every day. We're talking about VDNF. There are some special receptors. They are responsible for the survival and adaptation of the cell. Let me draw your attention to something here on this slide. You can see all the VDNF studies here. Atypicals, typical schizophrenic models, and postmortem studies. BDNF in the hippocampus region, whether there's any effect, decreased, no effect, increased, etc. So you can see that there's no consensus. Immune system and cytokines. It may lead to neuroplasticity or neurotoxicity, and in time, everything that starts with calcium activation will define the fate of the cell. This is the complex structure. And maybe you would ask why I'm showing you all this, and here you can see that all the neurotransmitters, receptors, mechanisms, everything is in interaction. And we are trying to understand everything, and we are trying to prescribe our patients. You're trying to create a prescription specific to that individual patient, but I am sure you're not always certain that you're prescribing the right medication. Every day new molecules are coming. There are also many side effects you have to take into consideration. I hope that we are on the right track.